Our first speaker is Dr. Michael Levin. He is the Distinguished Professor and Vannevar Bush Chair at Tufts University. He received his PhD in genetics from Harvard. He's currently, his current lab studies unconventional embodiment of mind using a combination of developmental biophysics, computer science, and cognitive science. Applications of the work on the collective intelligence of the body cells range across birth defects, regenerative medicine, cancer, synthetic bioengineering, and artificial intelligence. So we really are very glad to welcome Dr. Levin. He's going to be speaking to us from Zoom.
There are now known molecular biophysical mechanisms, specifically bioelectricity, that underlie the intelligence of body tissues. Now, this idea of the body having some sort of cognition that's distinct from the main mind of the individual was something that figured fairly prominently in alternative therapies of different kinds, but it was by mainstream science thought to be kind of a fanciful fairy tale, a metaphor that doesn't have a useful underpinning. And I'm going to tell you today that that's actually not true, and that we now have techniques to interact with and actually exploit this somatic intelligence for biomedical purposes. So Fabrizio Benedetti, who studies various kinds of placebo effects and things like that, has this wonderful quote where he says, "Words and drugs have the same mechanism of action." And if I came here and I told you that just with the power of my mind alone, I could drive sodium and potassium ions to depolarize 30% of my body cells, right? I could just by the power of thought, I could exert this molecular change in my cells. Now most people would think that's crazy, and a few people would think, well, maybe this is some sort of mind-body medicine, something to do with meditation practices, yoga, maybe some sort of biofeedback. But actually, this is something that happens in your body 24/7. In order for you to get up out of bed and go to work, you have to be able to take the high-level executive decision that that's the behavior that you want to do, and that has to feed down and control the depolarization of your muscle cells. So the entire somatic architecture is built on this ability to transduce from some level, whether conscious or subconscious, some level of intelligent problem solving, goal-directed behavior, and so on, down to the molecular qualities of cells and tissues and so on. And so the talk that I'm going to give today does not use any kind of unusual, let's say for physics, any kind of unusual forces, energies, nothing like that. I'm going to tell a story of very conventional physics underlying this remarkable continuum between mind and body, and that's possible because of bioelectricity. And so specifically, we have this spectrum here, which allows us to understand how very different kinds of systems, from simple machines to some kind of cybernetic kinds of things like homeostatic devices, to learning systems, to rational linguistic systems, all along this pathway. There are different degrees of intelligence, different types of tools that you might use to interact with these systems. And one question you might ask is, where do cells fit in, or more specifically, collective of cells in the body? Are they the kind of simple machines that are to be controlled by rewiring, or might they be actually amenable to various other techniques that we normally reserve for cognitive science, behavioral science, things like that? And the reason that it is possible to use these techniques in the actual medicine of human health is because we are made of a multi-scale competency architecture. So each level, from the molecules, the subcellular structures, the cells, the tissues, and of course the organism, and even groups and swarms in biology, are made of components that have their own agendas. They solve problems in various problem spaces. And so we have this architecture where every level is competent, and they're all competing and cooperating with each other to implement the thing that we call the entire organism. And this is because we are all made of a kind of agential material. We're not made of passive materials. We're made of this kind of thing. These are cells. This happens to be a free-living organism known as lacrimaria. You'll notice there's no brain, there's no nervous system, but it's very competent in solving its local tiny little problems as far as metabolic needs, physiological needs, behavioral needs, and so on. And so we are made of cells that have all kinds of internal capabilities. And the state of the art in molecular medicine today is that we're very good at this kind of thing, figuring out which genes and proteins interact with which other genes and proteins, so making this kind of map of the hardware. But we're actually a very long way away from control of large-scale form and function. So what we would like is a kind of definitive regenerative medicine where we are able to communicate anatomical goals to groups of cells and get them to build whatever we want them to build. So that would be healthy organs, whether in vitro or in vivo, addressing birth defects or traumatic injury or cancer. We would like to be able to communicate these large-scale goals, not just control this low kind of molecular level interaction, but actually be able to say that you want to rebuild a limb or make a new kind of biological construct or whatever. And so biomedicine today, I will argue, is largely stuck where computer science was in the 40s and 50s, where you can see what she's doing is she's physically rewiring the machine, and that's the only way that we had back then of interacting with these systems. And right now, all of the excitement of molecular medicine is all around genomic editing and CRISPR and protein engineering and things like that. And this is all at the hardware level. But as we know from the information technology revolution, much of the excitement is to be found in the software. And so this is what we need to understand, the higher level intelligent software that operates in our cells and tissues. And what I mean by intelligence is William James's definition, which is the ability to reach the same goal by different means. And this is important because it doesn't talk about brains, it doesn't talk about any of the details of whether you're a natural or an engineered system. It's a very cybernetic definition around this idea of having specific goals in some problem space and having some level of competency to find it. Now, we as humans are not very good at this. We have some capacity to recognize intelligence of medium-sized objects moving at medium speeds in three-dimensional space, crows and primates and maybe whales and things like that. But actually, there's intelligence in many other spaces, including gene expression space, anatomical morphospace, and physiological state space. And in all of these spaces, just like three-dimensional space, living systems at all scales navigate these various spaces with the ability to get around obstacles and get certain goals met and respond to novel kinds of challenges in those spaces. You could imagine that if we had evolved with a sense organ that was pointed inwards, let's say to give us a primary perception of our blood chemistry, we would have no trouble recognizing our various organs as intelligent systems that navigate these physiological spaces. We simply aren't good at noticing this because all of our sense organs tend to point outwards into this three-dimensional space. So there are many capacities. For example, it turns out that gene regulatory networks and molecular pathways actually can do 6 different kinds of learning, including Pavlovian conditioning. So even the molecular networks in your cells have the capacity to learn from experience. And this is one of my favorite examples of intelligence in anatomical morphospace, which you can see here. These are tadpoles of the frog Xenopus laevis, and so here are the eyes, the brain, the nostrils, and the mouth here. And so during metamorphosis, they have to turn into frogs. And in order to turn this face into a frog face, all of the organs have to move, the eyes, the jaws, everything has to move. And it was thought that this was a hardwired set of movements, that genetics provides each organ with some mechanism to move the right amount in the right direction. So we decided to test that hypothesis and see how much intelligence there is. And so we created what are called Picasso tadpoles. So everything is scrambled. So the eyes on the back of the head, the jaws are off to the side, everything's in the wrong place. And it turns out that these animals make pretty good normal frogs. And that's because all of these organs, starting off at abnormal positions, will continue to move in new unnatural paths in order to get where they're going to make a normal frog face. In fact, sometimes they go too far and they have to double back. So they have the ability to navigate this anatomical space of possible configurations to get from this starting position to this position, but also from this one. And in order to do that, you have to be able to do 3 things. You have to be able to recognize your current position in that anatomical space. You have to recognize the target, where are you going, and you have to be able to tell the difference and how to reduce that difference. So what genetics actually gives us is a very clever morphogenetic kind of system that is an error minimization scheme. It's a flexible problem-solving kind of system. And so that raises a very important question: how does it remember what the right pattern is? And could we learn to communicate with this cellular collective in various biomedical contexts to get it to go to the right place in morphospace? That requires us to know how it decides where to go in the first place. So we've been studying this now for some time, and what we've been doing is taking a cue from how brains implement goal-directed navigation, and they do this via familiar hardware in your nervous system, so ion channels and electrical synapses that allow information processing through the network of neurons that you have. And so here in the zebrafish, you can see this group made this wonderful video of all of the cognitive processes that are revealed by tracking the real-time living physiology in this brain. And you can try to do the sort of decoding to understand how can we infer the memories, the preferences, the behavioral repertoires from this physiology. And it turns out that every cell in your body does this. This is not just for brains. So all cells have ion channels. Most cells communicate electrically with their neighbors. And as a result, you can try to do the same kind of decoding and encoding research program to ask, what are the rest of your body cells thinking about? And it turns out that very much like in the brain, bioelectricity serves as the cognitive glue that binds together individual neurons into the collective you. The same thing happens in the rest of the body. The individual cells are bound together by an electrical network into a collective intelligence. And what does that collective intelligence think about? It thinks about body shape. So I want to show you some techniques that we've developed. So this is a voltage-sensitive fluorescent dye. So here, this is an early frog embryo, and you can see all of the electrical conversations here that the cells are having with each other as the collective decides where the head is going to go, the tail, the left, right, and so on. And this is a time-lapse of the electrical imaging of a frog embryo putting its face together. Here's one frame from that video. You can see already, long before the genes are turned on to pattern the face, you can see this is the memory of where everything is going to go. Here's where the eye is going to go, the mouth, the placodes, and so on. And this is literally an electrical pre-pattern, that subtle energetic scaffold that tells the cells where the different organs are going to be in the future. And we have lots of work that I don't have time to tell you about now, investigating this as an actual memory. So more important than just tracking these things, we've developed tools very parallel to what is used in neuroscience to control these electrical patterns in living tissue. So not just to read the memories, but to actually write new ones. And so the reading is done with these voltage reporting fluorescent dyes. The writing is done with techniques like ion channel modulating drugs, guided by a computer model that tells us which channels to turn on and off. Optogenetics, or light-gated ion channel activity, engineering of these channels to have new properties, and so on. Very important. There are no waves, no fields, no applied electrodes, no frequencies, no magnets, no electromagnetic fields of any kind. What we're doing instead, this is not applying any kind of exogenous electromagnetics. This is hacking the natural interface that cells are using all the time to tell each other what to do, right? So the reason all this works is not because we're all that clever, it's because we have access, we've gained access to the natural, very powerful interface that this cellular collective uses to bind individual cells to common purpose. And when you do that, I'm going to show you very briefly some results that you get when you start to manipulate this pattern. The first one has to do with organ induction. And so when we use, for example, ion channel RNA to put a little special little voltage spot that corresponds to that the eye spot in that the electric face that I showed you a moment ago, somewhere else in the embryo, for example, in a region that is normally made of gut, and so it forms the digestive system of this animal. If you give those cells that particular voltage pattern, that message is build an eye here, and they happily build this eye. If you section, these eyes can be quite normal with all the same lens, retina, optic nerve, all the right components. But notice that we didn't tell these cells how to build an eye. This isn't micromanagement the way you would with a passive engineering material. We didn't have to talk to the stem cells and tell them what to do and say where they're all going and all the different complex structures inside the eye. We gave the system a simple top-level command, build an eye here. And the reason that works is because this is a highly competent system that has the ability to build structures and then stop when they're done if it is told what it is that they should build. In fact, they have other competencies, like if we only induce a few cells to be an eye, they actually recruit a bunch of their neighbors. So the blue cells are the ones that we turn to the right voltage. They themselves convinced all these other cells to participate with them to build a lens of the right size. That type of recruitment, which by the way you see in other collective intelligences such as ants, but that ability is inherent to the organism. It's not anything we have to engineer. That kind of intelligence, that problem solving is something we can take advantage of. The other story I want to tell very briefly is about limb regeneration. So frogs do not regenerate their limbs at this stage, and so if they lose a leg 45 days later, there's basically nothing, except that interestingly at the time of amputation, you see that the opposite untouched leg lights up bioelectrically at exactly the same location as this leg got injured. And in fact, by watching these signals, you can tell where the injury was and what kind of injury it was. So this kind of information is potentially available throughout the body. It is absolutely not local to the site of the injury. But better yet, when we use a drug cocktail that we developed that changes the electrical properties of the wound and basically says to the cells, go down the regeneration path, not the scarring path, what you get is a turn on of pro-regenerative genes such as MSX1, and then immediately leg growth. And so you get by 45 days, here are some toes, here are some toenails. In our latest work, we've shown that a 1 day, so 24-hour application of this wearable biodome, this device, together with the ion channel cocktail, induces up to about 1.5 years of leg growth. So you don't touch the animal during that time. All it is communicating with the cells at the very early time point that says you should regenerate the leg. In fact, even more so, what it actually says is regenerate whatever normally grows here. You don't even have to tell it has to be a leg. And then for 1.5 years, it grows. You're not micromanaging it at all. And I have to do a disclosure here. We have a company with Dave Kaplan called Morphoceuticals, which is trying to bring this technology to mammals and hopefully eventually to therapeutics. But the idea is to use bioelectric states to convince cells to regenerate complex organs. And the final story here that I want to tell in this multicellular context has to do with birth defects. And so what we've shown is that very nasty birth defects, such as, for example, these kind of brain defects. So this is what a normal brain should look like. This is the brain of an animal containing a mutation in a key gene called Notch, which is an important neurogenesis gene. And you can see there's no forebrain, midbrain, and hindbrain are basically a big bubble. Turns out that if you use the computer model to ask how would we restore the normal bioelectric pattern despite the fact that this thing has a mutation, we've also done cases of teratogens such as fetal alcohol syndrome modeling with alcohol and so on. So if you ask the model what to do, it makes suggestions that then you can use directly to pick drugs, and you can pick drugs to override various teratogens like nicotine and alcohol, or even these genetic mutations. And here they get their brain structure back, they get their brain gene expression back, and they get their IQs back. They're back to normal learning level for these animals. And so the idea is that in some cases, and I'm not saying it's going to be all cases, but in some cases, you can even fix hardware defects, that is, defects derived from genetic mutations, by this kind of intervention at the physiological software level. And all of this is now being used to make a system, and you can play with the first parts of it here. It's all online, to make a system to pick specific tissues, whether normal or transformed, and figure out what channels are potential targets, and then the simulator will tell you how to get from the incorrect path to the correct path. And so lots of physiomic kind of profiling is required, much beyond the kind of transcriptomic and proteomics that we have now. So the last thing I want to mention has to do with cancer, and this wonderful thing that evolution has done is to exploit bioelectricity and other types of signals to scale up the cognitive light cone of individual cells. So this cell has a very tiny cognitive light cone in the sense of the scale of the goals that it's able to pursue. All of the goals are basically very local, very cell level. The rest of it is environment, and it will do everything it can at the expense of that environment to fulfill its local goals. So proliferation, certain metabolic goals, and so on. And so what bioelectricity and other modalities allow these things to do is to merge together and greatly expand their cognitive light cone to the point where this cellular collective has the goal of making this limb. This is a salamander limb. If you amputate the salamander limb anywhere along the axis, it will regrow exactly what's necessary, and then it stops. That's the homeostatic a goal-directed behavior, that journey in morphospace from here to here, and then it stops. Now, no individual cell here knows what a finger is or how many fingers it's supposed to have, but the collective absolutely does, because if you deviate it from that goal, it will do its best to come back. This is that William James's definition of intelligence. And so that process has a failure mode, which is cancer. This is human glioblastoma. And so what we've shown is that by injecting human oncogenes into these animals, not only can you tell, and so this is potentially a diagnostic modality, you can see the bioelectric patterns go wrong as the cells depolarize and disconnect from their neighbors. When they do that, they basically treat the rest of the animal as just external environment. They basically just roll back to their unicellular ancient past, and that's at that point they just go wherever life is good, and they migrate, they overproliferate, and that's metastasis and proliferation. And they're not any more selfish than normal cells. They just have smaller selves. And so now, given that framing, what you can do is try to force them to remain connected, not kill them as in with chemotherapy, but actually just use an ion channel to keep them electrically in the right state and connected with their neighbors. And you can see that here. This is the same animal, even though the oncogene is blazingly strongly expressed, and there's all kinds of oncogenes expressed here, there is no tumor, because it's the bioelectrics that drives, not the genetics. And we've started all this in the frog, and we're now in human glioblastoma and other cell types that we're trying to address. So I want to end here just by pointing out that biomedical interventions come in a couple of different flavors. All of this traditional stuff, these bottom-up techniques, really focused on the hardware, have made great advances, but also face a number of fundamental limitations. And there are top-down techniques, which we've only spoken about a few of them, that exploit the competency of the material. And so this is all given in these papers. And my points here are that future medicine is going to look like somatic psychiatry a lot more than it looks like chemistry. That bioelectricity is that interface layer that enables top-down control and communication with the collective intelligence of cells, and that we can now hack it for transformative applications in health. And I want to thank all the people who did the work, so the students and the postdocs and the technical support personnel, our many collaborators, our funders who support this work. Here are the disclosures of companies that have supported our lab, and most of all the model systems, because they do all the heavy lifting. Thank you very much for your attention.